Okay, guys, I think we're gonna start. 7:01. Okay. All right. How are we feeling about the exam? Good, kind of bad. Okay, nice. All right. So we're gonna go in order of like topics as you guys covered them. And if you guys have any questions, like feel free to just raise your hands. And we should stick around a little bit after. If you guys have any like specific questions, you can come see us. And this will be posted on the YouTube as well. So if you guys want to go back and watch it. Yeah.
And the slides will also be emailed to you guys.
Yeah. Okay. All right, we're gonna start with topic 1. Polypeptide anatomy. And a protein is just a sequence of amino acids. A polypeptide is like a sequence of amino acids. It's going to start at the N terminus. That's going to be your first amino acid, and then you're going to count up from there all the way to your C terminus. Our groups are arranged by chemistry and size and sterics. And so, like I was saying, primary structure of proteins are just the sequence of amino acids. It's like 1, 2, 3, 4. It's literally just the sequence in which they come in. These are held together by peptide bonds, which are covalent bonds, super strong and require a lot of energy to break. These are your strongest bonds in protein structure. Secondary structure is the backbone atoms. So these are largely composed of hydrogen bonds. These hydrogen bonds have like dipoles. There's a high difference in electronegativity between the atoms, and that dipole allows them to form hydrogen bonds with other dipoles. These bonds are not covalent, they're hydrogen bonds, and they can be arranged in an alpha helix. So there was like a problem on your problem set that asked about like the hydrogen bonds, and basically they'll be 4 amino acids away from each other. They'll want a hydrogen bond in an alpha helix. And then in a beta sheet, the R groups are kind of just arranged in an alternate fashion. Secondary structure is just those backbone atoms. It's not the R groups, and it's not those ionic interactions. For the tertiary structure, we see those R groups interacting with each other. These are composed of a bunch of different bonds. They have hydrophobic interactions, which will occur with your hydrophobic amino acids, which are your van der Waals interactions. You also have ionic bonds that will occur between positively charged amino acids and negatively charged amino acid. So ion-ion bonds. And then hydrogen bonds are also possible. So you can have dipole-dipole, or you can have ion-dipole. And then your only covalent bond in tertiary structure is going to be your disulfide bridges between cysteines. That's it for tertiary structure. And then quaternary structure is when you have multiple polypeptide chains. So it's not just one like sequence of amino acids. You're going to have 2 or more to have a subunit, a separate subunit. A singular polypeptide can have, we'll talk about like domains and subunits later on, but quaternary structure is only when you have 2 or more chains of polypeptides. Yeah, so tertiary and quaternary structure are not the same thing. Tertiary structure is just the interactions between the R groups. It's how the overall single polypeptide chain is going to fold. Quaternary structure, again, you have to have multiple polypeptide chains to see quaternary structure, and the same types of bonds are seen in both tertiary and quaternary structure. They are just not the same thing. Is that clear? And then you have domains and you have subunits. A domain will occur, like a domain will happen. It's just an independently folded section of a polypeptide chain. You can have this, you don't have to have quaternary structure to see a domain. You can have a domain on a singular polypeptide chain. For a subunit, these are gonna be the specific different like polypeptide chains. So if you have two polypeptides coming together, those will be separate subunits of the protein. Quaternary structure means you must have two or more polypeptide chains coming together. This is just like a little bit of an analogy of tertiary and quaternary structure. Your tertiary structure, if you think of it like a person, an independent person is one polypeptide, can have arms, legs, face, many aspects that makes that person, but those are the domains of the singular person. To see quaternary structure, you would have two people holding hands. The hand holding would be the chain by which they are connected, and that's the only way to see quaternary structure. Same thing for this train analogy. It's just same thing for the train analogy. One train has a bunch of functional domains, the wheels, the engine, whatever it may be, and then a series of trains would have quaternary structure, and each singular train in that would be a polypeptide chain. Yeah. Yeah. They'll also be on YouTube. So we'll also send them to you. Okay, so that was a lot of like structure talk. So now I have a practice question. A protein composed of a single polypeptide chain has been mutated such that a phenylalanine has been changed to an aspartic acid. Does this protein have subunits? And then explain why you think so. And then for question number 2, which levels of protein structure are subject to change with this mutation? And then why? So I'll give you like a minute or 2 to think about it and talk about it.
Like the lectures. I'm still giving them this. Like, super fast. I read it so much. Okay, but also like this is so like it's something from this topic more on your first exam. The trip off world? Yeah. Because when I took it, it was like this. Even though I took it, if I sit down, sorry. Not at all. I'll get up when it's got you.
If you have a protein subunit, and each of the subunits is just individually folded.
Yeah.
Would you say that the subunit is also like one domain, or would it have no domain? It's only like.
From that, you wouldn't be able to say like domain, like subunit is different from domain.
Right.
But a
Like-
subunit can have domains.
But if it's just one domain, it wouldn't really be a domain, because it's not in the.
Yeah. Mhm.
Okay, so that, in that case, you just see subunit.
Yeah.
Okay.
Like, I'm like, I need to draw things, but like, this is like one. So you're saying like, if you said one.
Yeah, so it's like 2 subunits, and each of the subunits is like individually folded from the other, but it's not like individually folded, it's not like makes it more.
Yeah.
Like, is that possible?
I think that could be possible, just like a subunit with no domains.
Like they're just, they're
They're just like, they're interacting with nothing.
Yeah,
Yeah.
or it's like they're both like just in one, like, they're both like scrunched up together. But it were two separate teams, like,
It's probably possible, but maybe if it's like a short polygon type shape, if it's really long, I imagine it.
right, so if it only folds once, it could be a domain, but it's usually not.
What do you mean folds one?
Yeah.
So like, you know how you drew like two kind, it was like clumped up. Yeah,
And it was like this.
yeah, so those would be two different stuff.
Yeah.
But if they were like clumped up like a domain, but they weren't, it was like on one study and it was just clumped up.
I think it just depends. And usually like in a question, she would let you know. But
So that's just like,
yeah, yes. And it's like hard to tell when there's a domain, like usually there'll be like specific domains, like kinase domains, or like they have like functions.
Yeah.
But just, I would just remember like, definitely you see quaternary structure when you have like those two separate polypeptide chains. Sorry, that was like.
confusing, but that's it.
Okay,
Thank you.
yeah, of course. Okay. Any thoughts? Anybody want to share? Yeah. Yeah.
Firstly, I believe that it would not have, it's not that many subjects, the single polypeptide, ones that help me there.
Yeah.
Also. Actually.
Okay. Yeah. Yes. Yeah, that sounds right. Okay, so for number one, like we had shared, this protein would not have any subunits. It's just a single polypeptide chain, so you just wouldn't see any subunits. Possible it would have domains, but we don't know that just from what was given to us. And then for number two, the levels of protein structure that are subject to change, I would say primary, definitely. You're literally changing one of the amino acids in the sequence, you're changing primary structure. I wouldn't say so much secondary. Those are the hydrogen bonds. You don't really see our group interactions there. So sure, like it might be possible, but it's unlikely. That's how I usually answer questions in this class. And then tertiary is like likely to change because you're changing a highly nonpolar amino acid to a negatively charged amino acid. So those interactions that it's making are going to be completely different. Phenylalanine is going to make those hydrophobic interactions, van der Waals interactions, and then aspartic acid is definitely going to make like an ionic interaction. Yeah.
Speaking of amino acids, the professor has mentioned that we're gonna have some sort of sheet.
Mhm.
Is that gonna be determining their hydrophobic and hydrophilic homes, or will that be determining whether they're like charged or not? Because I've seen two different charts going over there, and I was like, this is really annoying, like, oh yeah, these ones are just exceptions, what the heck are they supposed to be then? Tell us what the actual point is.
Yeah, so you'll have the amino acid chart. It's like the one that has 4 quadrants and it says like polar, non-polar. Yeah.
I don't know how well everyone can see this, but it's the one with the yellow, green, purple, blue.
Oh,
That's what you'll get on the
that's
exam. Yeah.
What do you think would be like a good example of one that would come up where it would be a changing the secondary? Like, if you think of one that might come up, it's just like look out for when it's a mutation of backbone making, so we're doing backbone.
Yeah, I would say like there was a problem on your problem set that kind of like came to mind when you asked that. It's like introducing extremely polar things to like the environment that the protein is in. So like molecule like urea is super polar. So if you add high concentrations, like high amount of urea to the environment of that protein, it's likely that the backbone will be affected because they're just hydrogen bonds. So anything that's available to hydrogen bond is going to disrupt secondary structure. Yeah. I don't think so. I think, like I said, it's like unlikely that that would happen, but sure, like there's a little bit of possibility, and that's like good evidence for that little bit of possibility.
To add on to that, when you're taking the exam, I'd be weary of saying like definite, it would not affect this. Cause for example, if there's a size difference between the amino acids, maybe it can affect hydrogen bonding, maybe they won't be able to fold the way they would initially. So you could say something like it's unlikely, but it may because of this. Like you don't have to elaborate that much. But if you can, I would just not make anything a clear cut. This doesn't, this does type of thing, because it can always vary.
Yeah, that's fair. It's biology, everything has exceptions.
Yeah.
Yeah,
I wouldn't say definite it will or it wouldn't, just it probably won't, but it could because of this one factor, because like the size or something like that.
I'll do this right away. I can do it. Okay, so this is just like going into a little bit about polar versus non-polar molecules. The polar molecules are gonna unevenly share electrons. So in a polar molecule, you're gonna see a difference in electronegativity between atoms. In this molecule, our oxygen is gonna have that negative dipole. All that means is electrons are more likely to hang around the oxygen. That's the way I would describe it. That's how I do describe it. And then non-polar molecules, there's like an even distribution of charges, or like the full distribution of charges, like in a negatively charged molecule, that atom has a full negative charge. So it's going to participate in ionic bonds. Yeah. Like in this molecule to the right, even though chlorine is super electronegative, it's balanced. Not that that really matters in this class, but that's why it's nonpolar. Okay, so this was a question. What specifically determines how hydrophobic or hydrophilic an R group is based on its structure? It's just like some characteristics that makes up the R group. A more hydrophobic characteristic is carbon-hydrogen bond or a low difference in electronegativity between atoms. Like I put two like nonpolar amino acids here, and you can see that they're just carbon-hydrogen bonds, which are super nonpolar. They don't want to interact with water. That's making those amino acids nonpolar. And then a more hydrophilic characteristic is the presence of these dipole moments. So electrons being more likely to hang out around one atom is going to make it a dipole moment. So in trinene, we have that OH group. That oxygen is pulling those electrons towards it a little bit more. So it has that negative dipole on the oxygen. Does that sound good? Okay. And then these are just the types of R group interactions that you'll see with like based on the category of amino acids. This is something that I would say is like super important. You're going to see this throughout your exam probably. Your polar charged R groups are going to make ionic bonds. Positively charged. Yeah, positively charged will bond with negatively charged. And that's all you need to know for that. Polar uncharged R groups will make hydrogen bonds because of those dipole moments that we talked about on the last slide. And then your non-polar R groups are going to make those hydrophobic interactions, the van der Waals. They stick together, kind of like conceal themselves from an aqueous environment of the cell. This is just some of the interactions that we see in tertiary and quaternary structure. It's kind of like similar to the last slide that I was talking about. Like I said, the types of bonds that each amino acid is going to make is going to show up on your exam, probably. It showed up on mine. So I would be familiar with this. Just know that your charged R groups can make ionic bonds. Your uncharged polar R groups can make hydrogen bonds. And then your non-polar R groups will make van der Waals interactions. And then these are the only type of covalent bond that we see in tertiary and quaternary structure. It's very strong. It happens between the sulfurs on cysteines. These are unlikely to break and unlikely to just be disrupted by the environment of the cell at all. They're super strong. Like we talked about, covalent bonds are the strongest bonds that we see in this class. So just know that they're super strong and they only happen between cysteines. And then this is just a summary of what we talked about. This is how you'll know like what types of bonds two amino acids should make with each other. As long as you know the property of each amino acid, you should know which types of bonds they're making. And yeah, primary structure and secondary structure. No, sorry. Primary structure, you see those covalent peptide bonds, they're super strong. They are unlikely to break. Secondary structure, you see hydrogen bonds in the backbone atoms. And then tertiary and quaternary, you see covalent and non-covalent, a variety of them. Remember, the only covalent ones you see are your cysteine interactions. And then this is just like a visual of how the proteins fold. Your hydrophobic groups are going to want to stay together. They're going to want to be concealed. They don't want to be outside of an alpha helix, for example. They just don't want to interact with that aqueous environment. So they're going to be more concealed. Oh, yeah. We will post these or we'll send them to you like via email.
Also, just to add, be careful when you're reading the questions, see what kind of environment the protein's in. Like on the problem set, it was embedded inside, what was it? A membrane, and inside the membrane's lipids, so it's a hydrophobic environment. So in that case, the hydrophobic amino acids are right on the outside. So their hydrophobic amino acids are only aggregated in the inside of the protein in an aqueous solution, but they're not necessarily always in an aqueous solution.
Gotcha.
I can pursue it.
Yeah.
So next we're gonna go over protein modeling. I'm gonna just read all of them here, but the next slide will have the pictures that correspond with them. So first, it's the ball and stick / wireframe model. In the ball and stick, the atoms are represented by the balls and the sticks represent the bonds. So the pro of this model is that it shows every atom and covalent bond in the protein, including the backbone and R groups. But the downside of this is that it's very hard to discern the R groups from the backbone, just because it shows literally everything. And then the wireframe is just the sticks, all showing the atoms. And then in the spacefill model, it shows the shape of the protein. So it's going to show every atom in the space that it takes up. So yeah, the pro is that it shows the overall shape of the protein, especially the surface. And then the con of it is that it's hard to see the backbone. And then in the ribbon model, the pro is it shows the secondary structure really well, like the alpha helices or beta sheets. Con, you can't see the R groups. And then backbone, the pro, it shows the path of the polypeptide backbone, doesn't show the R groups, which is the con. And then it's essentially the skeleton of a protein. And then normally you will use more than one model to show a protein. Like it says, you can overlay models on top of each other. And then it's for the exam and just in general, it's best to understand why one is better than the other in a certain situation. And then, oh, sorry, I forgot that I'm not going to show them. And then these are just the models here. I can do it if you want, or do you want to do it? If you want, I think you wanted to.
Oh, sorry.
If you want, you could do it. Okay.
Okay, and now KD and ligand interactions. Why is my computer doing that? Okay, so protein ligand interactions. A ligand is just anything that a protein is going to bind to. And then just think of it like they're it doesn't always have to be a binding pocket, but there's like a site for binding. You're gonna have your binding site composed of various different R groups and chemistry and shape. And then you want your substrate to be able to fit in there, like physically, structurally, in terms of size, but also in terms of chemistry. Like in this picture that we see, think about what types of bonds are being formed, think about the properties of each amino acid and what types of bond that they're available to make. And that's it. Yeah, remember just like the properties of your R groups and what types of bond that they'll make. Your affinity is just how much the ligand and the protein is going to want to be together. High affinity means that they would like to stay bound to each other. Your affinity is going to depend on the number of bonds holding the two together and the strength of those bonds. Here, I have like ranked the types of bonds. This is also something that you should think about when you're going about like questions like this. You're going to want to know like which types of bond are your strongest and which are your weakest. And then this was something on your problem set too, like making sure you know which protein ligand would be strongest, would have the strongest affinity based on the bonds between the two. Yeah. And then KD, just think, oh, sorry.
Generally, how many bonds do you need to like have changed or remove entirely before you say the ligand is no longer able to bind?
I think it just depends on like the protein and the ligand. I don't think like you can tell just by like memory how many bonds would have to change for it to like dissociate. But I would definitely just know like if you have a charged amino acid interacting with the ligand and you change it to something hydrophobic, that's going to change like the chemistry of the binding, which is going to change the affinity for each other. So it's going to decrease affinity. Does that make sense? Yeah. 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 Yeah.
Is that maybe is lower, right?
Yeah.
What is the angle of the bonds need?
I wouldn't worry too much about the angle of the bond. You're just gonna want to be concerned with the types of interactions that are being made. Do you have anything to add about angle bond? It's like super
think-
chemistry.
Maybe instead like the size of the amino acid,
Yeah.
like if it changes from a smaller amino acid to a larger one, that type of thing, or vice versa, that can affect the affinity. But yeah, I don't think you actually need to concern yourself too much about things.
So yeah, the lower the KD, the higher the affinity. KD is just your dissociation constant. So how often these molecules are going to dissociate from each other, or how long it's going to take for. The KD is the concentration at which the. Hold on.
If something was,
Yeah.
Oh.
But the lower the KD, the higher the affinity. Your KD is the concentration of ligand when the protein and the complex are equal to each other. Don't worry too much about what like that means, what KD means. Just always remember that KD is the dissociation constant and that when it's lower, affinity is higher. That's all I would concern myself.
okay, next, we're gonna just quickly go over enzymes. So, enzymes are needed in the cell to make and break covalent bonds because covalent bonds are so strong, sorry. It's gonna take a lot of energy to break them. So without these enzymes to catalyze these reactions will take a very long time. And as I'm sure Laura has emphasized in the class, the cell is a very dynamic environment. So sometimes these reactions need to occur faster. And then it's important to note that the cell has a mechanism of regulating these enzymes, so it gives it more control. And then an important one is ribozymes, and these enzymes are made up of protein RNA, and it's important to remember that it's the RNA itself that gives it the catalytic ability. Okay. So how do these work? It lowers the activation energy required for a reaction to occur. And it also can be referred to as a transition state because it's in between the reactants and the products. Enzymes lower the activation energy of chemical reactions, and you don't need to know in depth exactly how, but it just changes like the microenvironment, and that's kind of how it works. Like it doesn't change anything with the products or the reactants themselves. So that's actually the next point. Enzymes only affect the activation energy. They don't change the amount of energy in the products or reactants. Ideally, enzymes and reactants should have high affinities, while enzymes and products should have low affinities. So a good affinity with reactants ensures that it won't dissociate before any changes. And then a bad affinity with the product is best, so the product can easily dissociate away. So like, for example, if it had a good affinity with the products, then it would just stay bound, and then eventually another, like it couldn't react with another one to make whatever reaction it's doing occur. Okay. Next is protein trafficking. In eukaryotic cells, proteins are made in the cytosol by ribosomes. These ribosomes can float freely in the cytosol, or they're attached to the membrane of the ER. Proteins bound for the endomembrane system are made by those ribosomes that are outside the ER. So that's kind of like the co-translational aspect. And then proteins that go to the endomembrane system are either going to the ER themselves, secreted outside the cell, the lysosome, or the Golgi bodies.
You know, I've actually had a question. I remember a picture similar to this. It was one of the ones before. Like, I remember this picture of the endomembrane system. It shows the technology that might have had a connection to it. Like I saw 4 things that we ended up on the. I'm not sure if it was from like the alternate memory system or something, but it's the one where we combined contrast, chloroplast, and cytosol. It was.
Is it the peroxisome maybe? I think that was
Yeah.
I think that's also part of the endomembrane system. Yeah, it wasn't listed on here, but I remember that almost that question.
Yeah. Is it part of the endomembrane system or no? 'Cause
I believe it's not. It's not.
Yeah, because.
Never mind. No, it's not.
But is it like it went to the ER or something? Like, I don't know.
We
Fine,
can
let us know.
repeat the
It said
question.
that like, no, apparently, that peroxisomes who apparently have proteins that could go into ER, which confuses the heck out of me. It's a clearly from the graphic show that do not have it part of the endomembrane system. I don't know.
If you learned that in a different class, then I wouldn't worry about for this one, just focus on like what Laura says for us. Yeah. Okay. And then proteins that don't need to go to the endomembrane system or excreted outside of the cell will be made by the ribosomes that are floating freely in the cytosol. So these include solid proteins, so just the cytosol. Proteins that belong to the nucleus, mitochondrial proteins, and chloroplast proteins. And then this is just a diagram of ribosome in the cytosol versus one on the membrane down. Oh, sorry. Can you go back?
Oh, yeah, sorry.
Sorry. So a little bit about the endomembrane system. It's a group of membrane-bound organelles that work together to modify the package and transport lipid and proteins. And then composed of membrane-bound compartments such as the ER, Golgi, lysosome, and cell membrane. And then trafficking of these proteins through the system is called the secretory pathway. And then movement of these proteins in the system always starts in the ER. So you'll need, for any protein to either stay in the ER, stay in the Golgi, etc., it needs the ER localization sequence. And then to stay in that specific membrane, you'll need that retention sequence. Unless it's getting secreted outside, it lacks a retention sequence. Okay, and then just a little bit about signal sequences. It's a stretch of amino acids embedded in the primary structure of the protein. Sometimes it's at the very beginning, like with the ER localization sequence, it's in like within the first few amino acids. Sometimes it could be further in, like with the NLS.
Are we gonna be expected to know like exactly like sort of like the characteristics of the signal sequence, or is that gonna be like one of the charts that we're given as well? Just
Like
no.
specifically which amino acids do you mean?
Like the signal sequences, like for the different locations, like are we expected to know like, okay, like ER mostly has like a hydrophobic chain in
I don't think so. When I took this class, I've never been tested on like it has a hydrophobic chain.
okay, it's not like the actual characteristics. of
Yeah.
the okay, thank you.
Right.
Yeah, no,
Okay.
I didn't have anything like that.
Okay.
either.
And then like I said with blast side, every time it goes to the ER, it must have an ER localization sequence.
I have another
And
question.
then,
So it says newly made proteins are shipped off to the ER, but I thought proteins that were still being made by ribosome and finished in the ER, they had a ER localization sequence.
Yeah,
yeah, that's correct. That's probably just an issue.
I think like maybe they mean, maybe it's just like, we mean like newly made, like it's in the process of being made, but you're right. That's how it happens. Yeah.
Mhm. Okay. And then I think this is also on the problem set. The ER signal sequence is recognized by SRP, which is signal recognition particle. I wouldn't worry yourself too much with memorizing the name of it, but just know that it binds to something that brings it into the ER. And then if it doesn't have an ER signal sequence, it will not leave the cytosol. Yeah, that's basically something. Yeah. This is a lot here. I'm not gonna read through everything, just kind of the main points. But this is just like a little bit deeper on signal sequences. So, signal sequence can act as a binding site for other proteins whose purpose is to aid as like move the original protein to its intended location. So like the example, which we'll go over more in depth later, is like important and exporting. So, important binds to the localization sequence for the nucleus, exporting the exit sequence. That's just kind of what that's going over. And then typically proteins will enter organelles unfolded. So that's like with the ER. And then let's see. Nuclear proteins contain a signal sequence that resides on the exterior surface of the folded protein. And it kind of just makes sense because how else would like important or exporting bind to that protein if it's not on the outside? Like if it's hidden in the hydrophobic core, you won't be able to access it. And then this one, I've never been tested on it, but proteins need to be unraveled as they go through the organelle and then they refold once they are in the ER. And then, the video's not in here, but it'll be linked in the slides if you'd like to watch. And then the next few are also videos.
Yeah, this is,
Okay, so here are just some similarities and differences between the signal structures. So, with the ER and mitochondria, that localization sequence gets cleaved after entry. Whereas for the nucleus, it doesn't, it remains. Let's see. For the nucleus, oh, yeah. Yeah, I think that's why, I don't really think you're exactly tested on
It's
that.
very like, it's super, yeah, don't
Yeah.
worry too much
The
about it.
slide's
You
just
should know.
very Wordy, but
Yeah, it's
yeah.
co-translational.
Yeah.
Yes, but think
as
like
long as
that.
you know that, that's pretty much it. For the
Just
ER.
know that the fully folded protein in its like mature state won't fit through the channel. That's all you should know.
In the nucleus, the signal is part of the folded protein. And then for the ER mitochondria, it's a signal sequence embedded in the primary structure. And for this, actually, we could connect the nucleus, how it's part of a folded protein, to domains. So like, for example, that folded part of the protein is a domain. It has like that specific function, and that's what binds to important or exported. Let's see. Okay, nucleus, the protein enters fully folded and fully made. And then for the ER and mitochondria enters unfolded, and then the chaperone proteins help it refold. Nucleus signal sequence is on the outer surface of the protein. For ER and mitochondria, it's located in the end terminus. And then here's just a quick overview of the types of signal sequences, just condensed. So ER import and terminus, hydrophobic amino acid chain, followed by interspersed charged R groups after that. Like I said, I don't think you need to know in depth, like what the amino acids are or anything. ER retention, it's on the C terminus, which tells the protein to remain in the ER. And specifically what part, like Golgi, doesn't have it, it leaves it gets excreted outside the cell. The NLS, the nuclear localization sequence, it's where important binds, and it's on the outside of the protein. Export sequence, where exportin binds. And then, yeah, for cytosol, it must have an ER localization sequence, but will lack any retention sequence after that. Okay, and then here's a little practice question. A protein in the lab has an ER retention signal sequence. You decide to mutate it and add nuclear NLS. The first part, how will you see where this protein ends up? And two, where will you see this protein? And we'll give you guys a few minutes to answer that.
Honestly, can you change? Yeah. Can you change? ER, nucleus ER. I just made a typo. Here. The ER, sorry, I got it wrong. I don't even think it's gonna change on here. So I'm just gonna have to say- It might. Here, wait, let's see if I'll test it. If you want to exit it, it's still back in. I'm gonna do all that. I just made a typo. It happens to the best of us. Or maybe I should, right? So I don't confuse them. Maybe. I think it's, yeah, it's there. You just have to like go. Oh, oh, oh.
it called? GFP? That's what the. No idea. Yeah, it's like the name of a green.
Oh, yeah. Oh,
Oh, okay. Um. Yeah. Would anyone like to share what they thought for the first part?
These, green fluorescence protein or bacteria.
Nice. And do they want to share what they got for the second? Yeah.
What did you say for the first one?
You would use GFP, which is the green fluorescent protein, to tag it. Anyone for the second? I understand why you would think that this question, I think what it's lacking is it also does include an ER localization sequence. So with that in mind, does anyone else want to share what they thought for question number 2?
It would end up on the outside of the cell. Would be secreted.
right. Why?
Not quite. Does anyone else?
In the ER, for the most part, it has a good cycle into the nucleus, but it also might go back out, and once it's in the ER localization would be severed off.
Okay, so everyone has good ideas, but it will remain in the ER, but not because it'll come out of the nucleus and go to the ER. It's because the ER signal sequence is embedded in the primary structure, so it gets co-translated to the ER. Like if you remember the nucleus, that sequence is that folded part of the protein. So this will get translated as it goes to the ER, and since it has a retention sequence, it'll remain in the ER. Yeah.
So I thought proteins that, that was the ER had no other localization sequence. We just got to read that by itself.
So it has a retention.
1 second, they didn't done.
Can you repeat the last part of that question again?
Is this the type of question, what happens short term, or is that what happens?
It would be the long term too. If we're assuming that it has our localization sequence, it'll get co-translated to the ER. And then since it has the ER retention, it'll, once it's in the ER, it'll keep going. So it can't like somehow go back out to go into the nucleus or anything. But then since it has a retention signal sequence, it'll just stay in
It
the ER.
doesn't have ER as a sequence.
We're mutating it to add an NLS, but not change it.
Oh, it's that.
Yeah, it's just adding the NLS. It's not changing the retention to an NLS. Yeah.
But it's not mentioned if it has the ER localization, that's why it's, because it doesn't ER retention, it has the localization.
Yeah, that's just our bad for
Yeah.
not including it, but we're just
That
assuming
was
that it has.
like a,
the ER
sorry, yeah,
localization.
assuming that it does have the ER localization. That was an
Yeah,
assumption.
in the back.
Because it said you decided to mutate
Mutating doesn't necessarily mean destroyed. Mutating is just changing something. So we're changing it from not having that NLS to having it.
Oh.
Obviously, this question, you get the sequence there, that's the localization sequence. If you assume it has ER retention, then it has a localization sequence.
I wouldn't assume that. So anything's like ambiguous on the exam, you can always ask.
Yeah,
and someone will tell you, I'll let you know for sure.
yeah, this was our bad for not saying it has a localization. You could always ask, sorry.
Yeah.
That was on me.
If it just has VR localization signal sequence and you decided to retain and add the NLS, would the answer still be the same? Like, would it still go to the ER, or would it end up with this instead?
It would still go to the ER just because of that code translation, process.
And then
Like,
it
it'll get recognized by that particle in the ER, like the SRP, and then it'll get translated as it goes in and then I'll enter.
Cause I believe we had a question in class where it was like ER localization and then we added an NLS and then it ended up going to the or like vice versa or something. So I'm trying to figure out if it's like certain cases where it goes in and out or like.
Yeah, I mean, this was a question that I had when I took the class. It was like, it had a localization sequence and a retention sequence, and then you added like NLS. And then it was like retained in the ER. That is that was it. Yeah.
Thank you.
But like, for example, if we assume that it didn't have a localization sequence, then I feel like you could say you would believe it would end up in the units.
Yeah, if it didn't have
Yeah,
retention.
if it didn't have
Yeah.
that localization.
Yeah.
Oh, sorry, can you say that? If it lacks the localization, the ER localization.
Of retention.
Yeah.
Mitochondria and ER. I think that that's like super ambiguous, and I don't think that's something she would ask you. Just because those are like both embedded into the primary structure, so couldn't really tell which one would come first.
I
Would
would
be retained.
think it's whatever sequence is first in the structure, so it would be kind of hard to tell.
Yeah.
Yeah,
So is the reason that it's more so because of the fact that it has the retention signal sequence, or that would, I guess we're assuming, have the localization?
it's because of the localization sequence.
And the retention, yeah, so it won't leave the ER. So both.
That one would be hard to determine. She shouldn't ask you a question like that, because they both kind of go through the process of code translational. That whole, yeah, process. So I'd assume it's like whatever signal sequence is first, like closest to the N terminus. But there's not like a definite answer. The mitochondria also does go through a similar process. If you go back to the, what is it, the differences, similarities? It's not as emphasized as much, it's mainly the ER, just because there's like more possibilities, I guess, like you get secreted outside the cell. But, that's it. Because both of them, the signal sequence is leaved after entry, and they both enter unfolded. So. Any other questions about this practice question, I guess, or this whole section in general?
Yeah. Yeah.
Yeah. Yeah. Yeah. Even if there was no retention sequence, let's say it was just localization and NLS, sorry. It would still go to the ER first, but if there is no retention sequence, it would get excreted outside of the cell. Even if there is the NLS, just because it's not gonna go back in the cell. Any other questions? Yeah. It also does with the mitochondria, right?
Yeah. Just no, that for the mitochondria, for anything except the nucleus, it's not gonna go in fully folded. So for anything except like nuclear import and export, it's not gonna go in fully folded. So it's still gonna be unfolded, just because it won't fit through the channels.
Is that, do you have to see people?
I believe so, yeah.
Yeah. For this test at least.
Yeah. I think that's the only one she's talked about in lecture and videos.
Yeah,
So that's
it's the only one
all
you're
you'll be
expected
expected to know.
Okay.
If there's no other questions, we're gonna move on to nuclear import and export. So just a quick note, before I go through the process, you'll get both this import diagram and the export diagram on the exam, along with the amino acids. So you don't necessarily need to draw this on your cheat sheet or anything. You'll have the diagram.
I was actually really worried about having to follow that on the.
Yeah. Let me go for this exam. Okay. So for import, first important binds to the left on the cargo protein. Once it's bound to that cargo protein, it will enter the nucleus. Then RAN-GAP binds to RAN, causing GDP to dissociate, and then GTP diffuses into the binding pocket, and it becomes active. So it's important to note that for this, we just assume that there's an abundance of GTP in the nucleus, and that's just how it diffuses in. We don't need to know like how it recycles or anything like that. And then RAN-GAF, which is guanine nucleotide exchange factor, is a protein that exchanges GDP for GTP. And it's important to note that RAN-GAF along with RAN-GAP are not enzymes themselves. Only RAN is the enzyme. GAP and GAF just assist in the enzyme function. Where was I? Okay. And then once, sorry, where was it? Okay, next RAN GTP important complex will exit the nucleus. And then with the assistance of GAP, RAN GTP in the cytosol hydrolyzes GTP to GDP and important is released. So it does this because RAN-GAP has like an arginine finger, which stimulates RAN's existing rate of hydrolysis. It just increases it because without it, it'll just happen much more slowly. And then for export, RAN-GAF will bind to RAN, causing GDP to dissociate and GTP to diffuse into the binding pocket. So now it's in its active form. Then RAN GTP will bind to exportin, and exportin will bind to the nuclear exit signal sequence on the surface of the cargo protein. Then this complex exits the nucleus. RAN-GAP accelerates the rate of hydrolysis of GTP to GDP, which will cause it to dissociate along with the cargo protein. And what is it? Like I said before, note that GAP doesn't have that enzyme ability itself. It just helps RAN. And then RAN GDP has poor affinity for exportin, resulting in the release of exportin and the cargo protein. And then exportin is able to cycle back into the nucleus through NPC. Okay. And then a little bit more in depth, but kind of going over what I just said. GAFs and GAPs are proteins that regulate RAN during nuclear import and export. What is it? RAN is a GTPase. Oh wait, no, sorry. GAP is a GTPase activating protein, and RAN is a part of the GTPase family. I think that was like part of a question on the problem set. GTPase can hydrolyze GTP on its own, but GAP increases the rate. That's with the arginine finger. You don't need to necessarily remember it's arginine that does it, but just know that it increases the rate. And then, okay, the end result is that RAN is bound to GDP. GAP is needed for RAN GTPase to release export and import into the cytosol. And then GEFs bind RAND GDP and cause a change in conformation such that RAND can no longer have affinity for GDP. And then once GDP diffuses out of the nucleotide binding pocket, like I said, just assume that there's an abundance of GTP available in the nucleus, so it diffuse in unless otherwise stated in the question. Like, for example, she asked, what if there's a lack of GTP available in the nucleus? Then that won't be the case. Not in the nucleus, sorry, in the nucleus. Oh yeah, it says right here, GTP is at a 10 times higher concentration in the cell. And then once RAND's bound to GTP, it changes conformation, no longer has an affinity, and then the GEF dissociates. GEF is needed for RAND to be bound to GTP because this binding is required to remove important from the cargo protein. And this is just a little diagram of that process. And then before, I guess we get to the practice questions, anyone have any questions about import or export? Okay, so then here's just a practice question. How would nuclear export be affected if there was a loss of function mutation in RAND gap? And we'll just give you guys a few minutes. Yeah.
Can you go back to the last picture for a second?
Am I doing image measure?
Yeah, I'll do it.
Thank you so much.
Would anyone like to share what they thought for this question?
I think if there's a mutation in RAND gas, then the exportant would be unable to release the cargo. And it would most likely stay outside of the nucleus, meaning there wouldn't be enough export in the nucleus to get rid of the cargo. And they would just stay built up in the complex outside of the nucleus.
Yep, exactly. So what I had just written here, so RAND gap is responsible for increasing the rate of RAND's intrinsic catalytic ability. If RAND gap do not function correctly, GTP will take a long time to be hydrolyzed into GDP. This means that RAND GTP would remain bound to exportin, and the cargo protein would not be released. So over time, export will stop as all the exportin proteins accumulate outside of the cytosol and are unable to re-enter the nucleus and bring out more cargo proteins. Because if it doesn't, if if GTP doesn't hydrolyze into GDP, it gets remain bound to that export in, carbon protein complex. So it's when, it becomes grand GDP, it like loses its affinity for the import, or sorry, not import, export. Yeah, but the rate is just so slow that it'll just build up. Yeah. Any questions? Okay. And then this last section here, we'll be going over gene expression regulation. Gene expression is carefully regulated in cells at all levels. So just a brief overview of central dogma. In eukaryotes, transcription occurs in the nucleus, translation occurs in the cytosol. For prokaryotes, they don't have nucleuses, so transcription and translation occurs simultaneously in the cytosol. So DNA is read by RNA polymerase, which makes mRNA, such as transcription. Then the ribosome reads mRNA to know what sequence the amino acids should be strung together, translation. And then the protein folds into a specific conformation due to their amino acid sequence. So the R group is primary, not primary, secondary, tertiary, quaternary structure. So transcriptional regulation. This is the main site of regulation of gene expression. You have transcription factors without transcription. Well, there's two types of transcription factors. The first ones are what recruit RNA polymerase to the binding site, the promoter. Yeah, so without transcription factors, RNA polymerase cannot bind to DNA. And then you have regulatory transcription factors, which are gene specific, you have positive or negative regulators. So positive, it enhances transcription, so it increases the rate. Negative, it suppresses transcription. Okay, and then you have chromatins, which are just, it's DNA wrapped around histones. There's two types. So euchromatin is the open state. It's loosely bound around those histones, which you could see the bottom part of that image. And then you have heterochromatin, closed state, it's tightly wrapped around histones. So when it's tightly wrapped around, it's hard for transcription factors and RNA polymerase to get like recruited to start transcription, whereas in euchromatin, you see there's stretches of DNA that aren't wrapped around transcription factors, RNA polymerase, it all binds. Okay, and then there's RNA modification and processing. RNA has to be processed and made stable into mature mRNA to the nucleus. So how does it become more stable? You have the 5' guanine cap or guanosine and poly A tail. So it needs both of these things to exit the nucleus, otherwise it's vulnerable to nucleases that will like degrade it outside of the nucleus. Okay. And then just some more to become mature mRNA. So if you have introns, which are areas that are transcribed but not translated, exons that are both transcribed and translated. So to become mature RNA, the introns need to be spliced out. So that's done by the spliceosome. And then you also have alternative splicing, which allows cells to regulate which exons make it into the final mRNA transcript. And as so there's useful to get more diversity of protein products out of the signal sheet. There's also mRNA degradation control. An example of this is the sRNA pathway, which will go more in depth in the next topic after the exam, topic 4. For now, I would just know that that's an example of it. And then mRNA localization, cells can control whether mRNA gets localized to a new area. So that's like P-bodies. And when it gets sent to a P-body, it can either be stored there or it could be stored for later degradation. And then mRNA's lifespan can be regulated, and a cell has the ability to determine how long its life will be. Oh wait, there's still, sorry. And then there's translational control, which affects if the ribosome can bind. So an example is a protein can block or help a ribosome bind to the mRNA. Okay, there's also post-translational control. So allosteric, that was on the problem set. That's when a protein or something will bind to a site, not the active site, and it'll change the conformation of that protein. So for example, it can either hinder or allow it to function properly. So if it binds to an outside area and it changes its shape, and maybe the original protein that's supposed to bind can no longer bind, but also could do the reverse. Originally, this protein cannot bind the substrate. Once another one comes in, attached to a part other than the active site, it can open up and allow the original substrate to come in. So it's not necessarily always hindering or helping. It just depends on the role. And then post-translational modifications, it's just the alteration of a protein after it's been translated. Covalently added chemical groups, which can change the structure of a protein and therefore its function. So I think we went over this in 151 or 161, whatever you took. It's like phosphorylation, acetylation, methylation, etc. And then you have nucleotide binding. So like we went over with the import and export sequence, GTP and GDP can change the behavior of that protein. And then there's the degradation pathway, ubiquitin mediated proteolysis. Sorry if I said that wrong. I don't know if you need to know the specifics, but I remember from intro bio that it was 4 ubiquitins to tag it for degradation. Okay, and then here's just the chart of gene expression regulation. I would make sure you're able to describe, or at least with an example of each. I think when I took this exam, there was a question relating to these processes. So like, for example, transcriptional control, you have the chromatin, transcription factors, RNA processing, splicing, polytail, mRNA transport, localization control, talk about the P-bodies, etc. So just make sure you have an idea for each. Yeah.
What's a P-body?
It's just the structure that can either hold like the mRNA for a certain period of time, they can release it, and then it can go along the cycle, or it just holds it and stores it for degradation. You don't need to know like the exact specifics, but that's just like what it does. Well, transport and localization control. So anyone have any questions about this, or like unsure of an example of any of these? Okay. And then these are just some similarities and differences between prokaryotes and eukaryotes. So the similarities, both types of cells can regulate the degradation rate. They can regulate mRNA localization, the rate of translation, and then again, protein activity and degradation. But the main difference to note is that prokaryotes don't have a nucleus, so translation and transcription occur simultaneously, and that's why it doesn't necessarily have like poly A tail, guanine, black prime cap, because it doesn't have to worry about leaving a nucleus, and there's also less nucleases that will try and degrade this mRNA. Oh, also in prokaryotes, they don't package DNA into chromatin, whereas they do in eukaryotes. And in eukaryotes, there's basal transcription factors, whereas in prokaryotes, it's the sigma factor, which you'll learn more about in the topic after the exam, because that goes with like the trip operon and lac operon. That's it. Yeah, so don't have any questions, wanna go over anything? Okay.
I think if you have questions, we'll come to you or come to us. Good luck. You got this. That's it.
I know.